Well, welcome back. Again, we are here for our last session of the Smithsonian Education Online Conference, where we're focusing on problem solving with Smithsonian experts. We hope you've been enjoying the conference. If this is your first time joining us, I wanted just to make sure you knew how to interact with us. Over on the left side of your screen is a chat window, and at any point you are invited to send in your comments and your questions as we go along. And our Smithsonian colleagues here will have a number of questions for you as well, so we hope you will take some time to share your thoughts and ideas with us. So listen for those questions and do share. I also wanted to remind you that our session is being closed captioned, and we hope you find that useful. Thanks to our friends up at WGBH in Boston for doing that with us today. If you do not need the closed captioning, you can turn that off by clicking on the CC button on the right corner of the captioning area and simply change your display to none. But we do hope you find that useful. You might have also noticed that we have with us Dan Porter, who joins us from Kent. And he is logged in where it's a little bit later in his day. We have people from all over the world, but Dan is actually drawing and illustrating what he sees happening here in the session, and we'll be sharing those illustrations with you on the program page, and we'll also show you a few of them as we conclude today's session. So do stand by for that, and thanks to Dan for being a focused listener and such an imaginative artist when it comes to capturing what we talk about during our diversity track today. We're going to go ahead and get started. We'll have some closing remarks at the end, and we hope you can stick around for that. But I'm going to go ahead and kick things off by turning the floor over to CJ Geraci, who is going to begin our exploration of this big question: how and why do we count living things? Hi, CJ.
Hi, Jonathan. How are you?
Good. I'm glad you're here.
I'm glad to be here. So in the last talk, you guys learned about how some Smithsonian biologists study big vertebrates in the rainforest, and we're going to switch our focus and talk about really, really tiny invertebrates way up in the tops of the trees in the canopy. And we're going to talk with Terry and Michael about how we've used data from a conservation project to ask some questions about patterns of distribution of beetles and how that relates to the patterns of trees in the Amazon. And this is a project that's ongoing, so we don't have final answers, and we're hoping that you guys can give us some creative suggestions of how we could go about solving some of the problems that we've encountered during this project. Okay. So I'm going to start by showing you two pictures of two different insects that I encountered when I started working with Terry Irwin in 2006 in Ecuador. We were working near Yasuní National Park in eastern Ecuador in the rainforest. And these two insects are important. They were important to me because one of them I was down there specifically to try to find as part of my dissertation project, and the other one I was very much hoping to avoid. And so you could probably figure out that the one on the left, the big, giant, scary-looking ant, is the one I didn't want to encounter. This is the bullet or the conga ant, and there's probably lots of other names for them depending on what country you live in. But this is rumored to be one of the most painful stings in the insect kingdom. And I, luckily, have not been stung by one yet. Terry has been, right? Twice, yeah. So the insect on the right is in the order Caddisflies, or the order Trichoptera, which means hairwing. And this was the group I was studying for my dissertation. And I was trying to collect DNA from all the different types of insects in this one particular family. And this was one of the ones that I had yet to collect DNA from. So when I heard that Terry was going down to Ecuador, and I knew this species was found in Ecuador, I asked my advisor if I could go down there, and he was very nice and told me sure. So I went down there and was expecting to take all 6 weeks to try to find this specimen because I thought it was so rare, so elusive. And when I got down there, the first or second night we were in the field station, I walked up to the wall of the lab, and there it was, just sitting right there. So that's the specimen that I photographed and then put into a jar of alcohol and sequenced DNA from one of its legs. So while I was there, for the rest of the time, since I had fulfilled my mission of collecting that one specimen of caddisfly, I started paying a lot more attention to the beetles. And I became captured by just the sheer diversity of what I saw there and how beautiful and unique all of these beetles were. So I wanted to pose the question to you: why do you think we study insects? Why should we study insects? And do you think that insects are beneficial, harmful, or neutral? So I'll let you guys think about that for a few seconds. You guys have any suggestions?
It's a great question, and we've put a poll up, but we are also interested in hearing your responses. So go ahead, you can continue to type over on the left side of the screen.
Okay, so I'm seeing you guys switching back and forth between beneficial and both. Okay, well, yeah, I think the answer is both. It depends on what species of insect you're talking about. If you're talking about insects that pollinate fruits, they're of course very important. If you're talking about insects that transmit disease or insects that are pest species, then they're harmful. Some species could be beneficial in one ecosystem and harmful in another. I think that's what makes insects so fascinating, is that they are everywhere and that they fulfill so many functions, and we use them for so many things, yet we fear them for so many reasons.
CJ, we have an interesting point from Lisa. She says that insects are an indicator of the health of the planet. Would you agree with that?
I very much agree with that, and I think she's predicting what I'm going to say next. Maybe she's a little bit psychic. So the next thing I want you guys to think about are some direct and indirect uses to humans. So what are some direct ways that we use insects? Or some direct ways that they help us?
And
some suggestions.
Put that question there on the left side of the screen as well. Direct ways and indirect ways. We are hearing about pollination as
exactly. One. That's I think probably the most popular answer. Terry, you have the statistic of how many of our crop species are pollinated by insects.
No, I don't. But of course, all of the fruits are anything with a flower. But then a lot of our crops are grasses, and grasses are wind pollinated.
And of course, honeybees are a hugely important pollinator, and there's a lot of concern over the health of honeybee colonies right now. Another use, I don't know if anybody said this yet. Oh, I saw somebody say silk. Yeah, so anytime anybody wears anything with silk, this is of course from an insect. And there's also been some research done lately on the species of, again, the group of insects I studied for my dissertation, caddisflies, by my colleague out at the University of Utah, Russell Stewart. And he's looking at caddisflies and also a worm that lives, a marine worm that lives in the ocean, on using their secretions as glues. In the case of the sandcastle worm, he thinks that he could synthesize glue based on what this worm does to hold bones together during surgery. And so when I saw this article about the sandcastle worm, I emailed him and said, have you looked at caddisflies? And so he went out and collected some caddisflies in the Provo River. We're both fly fishermen, so we both knew where to find them. And he went and looked at the silk, and he figured out how the silk can be sticky underwater. So if you guys are interested more in these two topics, you can go to those links and read those articles. So I'm hoping to work with him more this summer to look at the diversity of the ways in which caddisflies use silk and how that might be useful for humans as an adhesive during surgery. All right, so those are what I think are some direct ways. This is another picture of what a capture net looks like. If you look up, let's see if I can find. Let's see if I can get my arrow. Let's see, do I have it? Oh, this one. Okay, bear with me. All right, so I've got my pointer. So this is the capture net that the caddisfly spins out of silk. So you can imagine knitting a sweater while someone turns a fire hose on you. That's what these guys do. They spin this silk underwater. And this is what the larva looks like. So this larva will sit right inside of this structure here. This is the retreat they make, and this will sit underneath a rock, underneath the water, and they'll spin this capture net. So again, the connection for me between caddisflies and the Amazon is that I was in the Amazon to look for a caddisfly and wound up being captivated by beetles. And what those things both have in common also is that we use insects in many different ecosystems, freshwater ecosystems and in the Amazon, as indicators of environmental quality and change in environmental quality, as indicators of, say, water pollution. And so this is a project that I've been involved with for several years with my colleagues from the University of Guelph in Canada. We're trying to build a DNA library of caddisflies so that we can help groups that are trying to monitor change in the biological community of streams and rivers. Changes in time due to things like water pollution and changes in water chemistry. And so this is very similar, actually, to what Terry was working on in Ecuador. So if we go to the next slide, we have the question, what can insects tell us about rainforest change over time? And so from here I want to turn the mic over to Terry so he can explain how this project came about and how he set up the original project.
Well, CJ, many years ago in the Ecuadorian Amazon, one of our oil companies here in North America really trashed the environment in the Cofani villages. So Ecuador was very sensitive to the next project of oil companies building roads across the Amazon, particularly right here on the edge of Yasuni National Park. People are very sensitive about what goes on by parks, but at the same time, 60% of the Ecuadorian economy at the time was oil driven. And so they had to seek out the oil. Well, what the government of Ecuador did was say, okay, you can build this road through the Guarani territory. You have to deal with the Guarani indigenous peoples, but also you have to have environmental monitoring on everything you do there. So having been a Canopy warrior and an Amazon scientist, actually having lived in the Amazon for 15 years of my life, I was a target for the company that got the contract to actually biomonitor everything that the oil company did. And so I was one. I had colleagues in fish, had colleagues in birds, in mammals, reptiles, even a social scientist that looked at the impact on the Guarani. So the idea was, as the road was being built, we had to monitor the impact on all of these creatures, and then over 3 years, the use of the road, so cars and trucks, oil trucks going back and forth up and down the road, there'd be an impact from that too. So then the next thing was to, well, we asked the question is, what impact did the road and its use have on the in my case, the canopy entomofauna, all the insects and their relatives that lived in the canopy. And each one of these scientists asked the same question about either the birds or mammals, but mine was the insects and their relatives. So our area, as you can see, the brown streak down the center of the green, that's the Andes Mountains.
Here's Quito.
And there's the capital, Quito. And our research area was out in the east, as CJ said, in the lowland rainforest. It's about 200 meters altitude. And if CJ drug that arrow all the way to the Atlantic Ocean, it would be 4,000 kilometers. That's the Pacific. And then all the way to the right-hand side, 4,000 kilometers down the Amazon River to through Brazil. So we're going to focus on two sites in the Yasuni National Park area where we carried out the project. We set up two plots, one right next to the road on the lower right, and the other is a control plot 21 kilometers into the forest that had no contact with the indigenous peoples. There were no roads, and it took 4 hours by canoe to arrive to our other site on a very windy river. So in each of these places, we set up a transect, and as you can see, there were 10 transects per plot, 10 samplings per transect, and that made 100 sampling stations for each of the plots. So we have 200 permanent locations that we were going to monitor the entomofauna of the canopy through 3 years, and then in 2005, 2006, we came back and did it again. So we had trans-seasonal, 3 samples per year, transannual, 3 years, and then transdecadal across 10 years.
Terry, a couple of questions just to clarify what a transect is.
We're going to show you a map in just a second.
Perfect. Stay tuned, Tim and Eric.
Yeah, so this is the plot scale. These are our 2 plots. That's the largest spatial scale. So here is a map of the plot. So this is the bottom. This is the road, this brown line right here, the road that they had built. And then this is our first transect, which is right next to the road. And then every one of these horizontal lines represents a transect that goes further and further into the forest. And so Terry, why did you build your plot like this?
Well, what we wanted to know was, is there an impact from the road building and its use? So our hypothesis was, if there is an impact, it will be more severe right next to the road than an entire kilometer into the forest. So this transect, as you see there, is 1,000 meters or 1 kilometer. And I set out to test that particular hypothesis. A blow-up of one of those transects is Transect 9, which is 100 meters across. And as I said in the last slide, we had 10 sampling stations along that transect. And if you look at it, the transect's sort of an oblique angle. You see how the trees are aligned, and the little white squares under the trees are sampling stations.
And this is a flat map that was published in a paper that you did in 2005. And so what our mission was when Michael and I started working here was, how do we turn this into an interactive database that we can use to answer questions and do queries? And so Michael's going to talk about that in a little bit. So we want to pose the question, if all of you were in charge of this project, how would you collect bugs from the tops of trees? Does anybody have any ideas?
I'm really curious to hear what people have to say. So you'd like to collect bugs from the tops of trees. What would you do? And we have some results coming in already. I am going to let each of you submit something before we let you start to see what your colleagues are sharing, so you can just think on your own a bit about this. Okay, we've got some results coming in. Okay, let's start to see what some of them look like. We have climbing and trapping. Somehow a butterfly net, or using a sticky bark-like substance. We're hearing people talking about climbing.
Those are all interesting ideas, and I think they've almost all been done. Except for the sticky bark-like substance. That's an interesting idea.
Well, that's, CJ, remember that's called sticky trap?
Oh,
and some of my colleagues have tied a rope on sticky trap, and on a pulley they pull it up into the top of the canopy and leave it there for perhaps a day or maybe a week, and then they let it go down, and then they investigate all the bugs that were flying and got stuck in the sticky trap.
And I've seen a few people say that they would set up a tarp underneath and see what falls down. And you guys are on the right track for what Terry did in this project. So how would you get the bugs to fall down into the tarp? That's the next question. What would you guys use?
Well, we've had a few answers come in on that. The students, there's some folks in Virginia Beach who said maybe using some kind of a bright light on the tarp. Some will just fall down on their own or shake them out of the trees. But we also had a group mention that perhaps they would use some kind of fogging substance.
Yay.
All right, you guys answered it. Let's see. So the canopy fogging technique was actually developed by Terry back in the early 80s. And so you want to talk a little bit about how you fog the trees?
It's kind of interesting. The first fogging was in 1974 in Panama, and a couple of my colleagues who are mammalogists studying anteaters wanted to find out the biomass of ants and termites in the canopy where these anteaters actually lived and fed. And they had a colleague there in the Panama Canal Zone who was in mosquito control. And he said, well, why don't you take this mosquito killing device that we use to fog backyards, and you could fog the canopy, and the termites and the ants would fall out. So that's how I got started. I was the only entomologist on that particular trip, and when I saw what came out of the top of the trees, all the beetles. Some of the beetles I only knew from one specimen in the London Museum, and I now had 5, 6 more specimens of that rare species. That was it. That changed my entire career, and I have been fogging almost every year since 1974. We use a biodegradable insecticide called resmethrin. It persists in the environment for only 2 hours before it breaks down, and because we move to different places all the time, insect resistance does not build up. And as I said, I've been working every year since 1974, and I'm perfectly healthy. And the thing is that the chemical does not affect vertebrates. And so I don't worry about fogging a monkey because it just doesn't affect humans or any kind of vertebrates. But it works particularly on insects and their relatives on their nervous system. And in fact, all of you who happen to live where you may have cockroaches in your kitchen, you probably have a can of Raid right under the sink that you'd use on the cockroaches. Well, it's the same chemical. And it was derived from a African sun daisy this particular flower in Africa has an insect-resistant juice. And so the people in chemistry extracted that juice, and now we use it for household stuff.
So this is a shot of, well, what our sampling sheets look like. So now we have 3 different terms. We have the plot, which is the impact plot versus the control plot. That's the largest spatial scale. And then within each plot, there are 10 transects. That's the next lowest spatial scale. And then the finest spatial scale are our sampling sheets or sampling stations. And this is what they look like. This is what they look like when we unpacked them from the box. So we spent probably about an hour trying to untangle them. And then the day before we fog, we go out and we set each sheet up in the forest. First we have to find the locations of where Terry had sampled before, and each one is 3 by 3 meters in dimension.
And it's easy to find the locations because when we laid out each of those plots, we put a permanent forestry stake at the center of the sheet. So for the next 10 years, every time we went, we just found the stake and put the sheet up in exactly the same place. So we had consistency through all of the sampling during the 3 intensive years, and then the follow-up year in 2005, 2006.
So here's some shots of the actual fogging. This is me learning how to fog for the first time, and it's done at 4 in the morning. You want to explain why that is? Why he makes us get up at 3 o'clock in the morning so we can get out to the plot by 4?
As soon as the sun, just the first rays of light peek over the horizon, it disturbs the microclimate or the air in the top of the trees. And so the fog will rise, but then it'll go off to a left or right, depending on which way these microclimates are pushing the wind. So at 4 o'clock in the morning, before there's any hint of light, the microclimate is completely still, and as you can see there on the right, the fog goes straight up into the top of the trees.
So now you want to...
Yeah, I was just going to point out the Amazon women there
we'll go
were
back.
my helpers.
Yes, so this was the group of scientists that I got to work with down here on the left, and it was really a fun group to work with, I have to say. It was an awesome 6 weeks to be in the rainforest doing nothing but collecting and sorting insects. And some people may think I'm crazy for saying that, but it really was cool. Okay, so after the insects fall down, we go back to the field station and have some breakfast and wait for all this to happen. We don't actually stand out there in the rainforest and watch them fall.
Quick question about them falling from Tanya in Cincinnati. How or why does the fogging make crawling insects fall rather than just sort of stop where they are?
As I mentioned, the action of this particular insecticide is, and I'll get a little technical here, but the way the nerves are in insects and their relatives is they have little synapses. So if you kind of put your 2 fingers together very close but not touching, then the nerve transmissions goes across that little gap in the nervous system, and this chemical actually interrupts the transmission of nerve impulses. And so what happens is they start shaking, and they just shake themselves right out of the tree, and they die about 5 minutes later by the actual chemical. So we have in the chemical something called a knockdown agent.
So this next video you're going to see is what we do when we go out a couple hours later. So this is probably about 7:30, 8 o'clock in the morning. And this is how we collect the samples from the sheets.
All right, we'll play a few seconds of this for folks now.
Okay. I know.
Por el uso. No,
And
oh.
this is the co-PI on the project. This is Cleopatra Pimienta from Colombia. So we're shaking the insects down, and they fall down into a hole in the middle of the sheet and into a jar of alcohol.
And the young woman that you saw there in the first part of that film with the red bandana is one of my Colombian students, Cleopatra. This is Ana Maria. She's from Ecuador, but the first one was Cleopatra. And she just happens to be right over next door in the Natural History Building now working on beetles from these samples.
Okay. So these are some shots of what we get from the sheets. You can see this is the bottle of alcohol that we collect insects in. Get my pointer back here. So here's the bottle of alcohol, and this is what one of the sheets looks like that has particularly a lot. It looks like a lot of grasshoppers in this one, and katydids. And then this is what we spend the next 6 weeks doing: is sorting insects to target taxa, and putting them in these vials. They start off in these big jars with the blue lids, and then we try to sort them into these little vials, and we're trying to pick out target taxa that we can then do statistics on.
A quick question before we leave the too far from the video. Terry was holding a stick in his hand. There was a question as to whether that was a blower of some kind, or what was that stick you were using?
These sheets, because of the humidity in the rainforest, and perhaps even it rained the night before, the sheets get wet and the insects stick to the sheets. So what I have on my back that you can't quite see is a garden sprayer, a 2-gallon garden sprayer, and we have 80% alcohol in there. And what I'm actually doing after the shaking, I'm washing the sheet with alcohol, and that goes down into the bottle, and the insects are preserved.
Okay, so here's another question for you guys. Why is it hard to count rainforest insects? Why do you think our next job after we get these samples is particularly challenging? And sort of a follow-up question to that is, how many insects do you think are in every hectare or 1 square kilometer of rainforest? I want you guys to take a vote.
So there's 2 questions there. You can answer both. So the poll is right in the center. How many insects do you think there are in essentially 1 square kilometer? And you can go ahead and vote as well. And then the question, why is it hard to count rainforest insects? We're hearing things like coming out of Erie, Pennsylvania, just the sheer diversity. They're very small, their size. There are just too many individuals and too many species.
Okay, those are all great answers. And
they're always moving is another one.
Until we put them in alcohol, then they don't really move much.
But they're happy.
But they're happy, yeah. Okay, so the answer to the poll, and this is based on some work that Terry did with some colleagues estimating how many types of species, different species, and how many individuals. And the estimate is actually at least 100,000 species per hectare and 3.2 times 10 to the 10th individual insects per hectare. So on the left you have a species richness number, and on the right you have a species or an individual abundance number, and we'll talk about those terms in a little bit.
So to understand that 3.2 times 10 to the 10th, you can go out at night and look up at the stars and count all of them, and you won't get that far.
Right, and so the only word I can think of to describe that number is gazillions. Okay, so this was our total sampling effort. These are the number of samples that we were able to take. We still need to go back to the control site and take 300 follow-up samples so we can do a full statistical analysis, and we're hoping to do that soon, pending funding. What we got out of that was 1,765,454 individual insects from our target orders, which are on the left-hand column. And these are only from the samples that were taken from 94 to 96. So our next question, what would you do with this many bugs if you had to handle them yourself?
And while you're thinking of that question, the reason we did target taxa is there's various reasons, but one thing is we couldn't possibly do all the families and orders of insects. And so what we did was take a random sample of 88 collections, and we counted everything. Well, as it's really interesting for those of you who are interested in ants, 50% of the abundance of canopy insects are ants. So half of our samples. So that means if you add all of these other things that we counted, it actually comes up to over 3 million specimens for the 9 seasons that we did in 94 to 96.
And they're all in my office. So I have 3 million office mates right now. So what we had to do with these many, this sheer number of insects was use them to come up with an answer for the oil company, and we had, 30 days to do it?
30 days after the field season, yes. We had to give them a report.
Right, so this is what we were able to do, and so this is a graph showing the change in relative abundance of just the insect orders. So just, beetles versus hoppers versus bugs versus bark lice. and versus ants and, well, wasps and bees, because we didn't count the ants. And so you can see that actually I think it's remarkable that the beetles only changed by, what, 0.06%? There were some changes in some of the other groups between the time when the road was constructed and 12 years after. So the question I want to ask you guys is, do you think this is a satisfying answer? Do you think this is the full answer? Do you think this is a correct answer? What would you do with this answer if you were a decision maker? Some things to think about.
So there's the question, and we're starting to get some responses. Thank you, everybody. And if you need to see that question again, Adam has put it up on the left side of your screen. We are also still getting some answers to your prior question, by the way, about what you would do with that many bugs.
Oh, okay.
There are people who are wondering if you display them, by the way, at the Smithsonian.
We're working on it, yeah. We're working on imaging them and putting them into a database, which is what we're going to talk about next.
Good. We're getting mixed responses, by the way, as to whether
Okay.
people think this is enough and it's conclusive.
Okay. Well, let's assume that it's not enough, that we want to give them a more detailed answer. One of the things we could do is look at a finer level of taxonomic resolution. And so this is a genus of beetle that Terry's been working on for how many years?
Too
40?
many decades.
Yeah.
I don't want to tell.
And so the reason you might want to look at a lower level of taxonomic resolution is because maybe even within the same genus, you have some insects that are very sensitive to some environmental stressors and some insects that are very insensitive to them. And so if you track the relative abundance and change in abundance over time, that can tell you something about sort of micro changes in the forest over time. And this is what we do with aquatic insects in aquatic ecosystems. So these are just some of the beetles that Terry has looked at, and some of the beetles that he has described from the genus Agra. This one was named after Sasquatch. This one was named after E.O. Wilson. And then this is the Terminator beetle that Terry named after Arnold Schwarzenegger. And we're being a little bit tricky here. These are actually Arnold's thighs superimposed over the beetle's thighs. Our graphic artist did that.
But the reason it was named Schwarzenegger eye is because the real beetle actually has those swollen femora on the middle legs. We only know dead beetles in the museum of this particular species group, so we don't know what they do with those legs. So that would be a very interesting study to find these alive and then figure out, are they jumping legs or do they lift weights? Who knows what they do with those kind of legs. But we did a Smithsonian Spotlight filming last year, and the producer and CJ and a couple of my others students sent this beetle picture to Arnold, and he signed it and sent it back, and they surprised me on the Smithsonian Spotlight and presented it to me live, well, not live, but during the filming of the thing, and my mouth must have just dropped. I opened this blue folder, it said from the Office of the Governor of California.
It was pretty cool.
That's hanging on my wall now.
Yeah. So now we have a vote we want you guys to take. Since Terry's named a beetle after Arnold Schwarzenegger, he's also named one after Liv Tyler, Kate Winslet, Catherine Bell. Do you think Terry should name an agri-beetle after Stephen Colbert? Let's see what you guys think.
And we'll, of course, report these results back to
Yes.
Stephen
Okay.
Colbert,
Yes.
won't we?
So if Stephen Colbert is listening, you can maybe expect a beetle to be named after you in the future.
Well, more than 80%.
Okay. All right.
I appreciate the people saying no.
Okay, so let's talk now about what we do with the beetles that we can't put names on. So the groups that don't have the world expert working on them at this time. So we have about 50 to 80% new species, we think, in these Canopy samples. And for many of the groups that you see here, these are all different families of beetles. There is no specialist, or there's only a few specialists in the world who know how to identify these. And if you guys want to know what families these are in, and if you want to guess and see the answers, you can check out the book chapter that we'll post on our virtual exhibit hall. Now that has the answers. So what we had just from the first few samplings is 300,000 specimens across 95 families of beetles. And we sorted these to what we call morphospecies. They're hypothesized species. We looked at them under a microscope and we sorted them into clusters that look the same. So, and we just give them numbers. And so these are our hypothesized species. And we can test those hypotheses later by examining the beetles in more detail under a microscope, maybe looking at the DNA. But right now we have hundreds and hundreds and hundreds of morphospecies that we think, to our best of our ability, are distinct and that we can use to do some statistics. Of course, the problem with that is just the sheer data overload. So this is just a collection of snapshots from all the Excel spreadsheets that we had. These are what the specimens actually look like when they're pinned with labels on them. And so you can imagine thousands and thousands and thousands of these records and thousands of specimens all in drawers. And so what Michael's going to talk about next is how we took all of these different types of data and put them into a database that we are still finishing building, a geographic information systems database to organize all of these.
All right, well, hi. It's the first time you're hearing from me. So what I wanted to talk to you about is using technology to turn the chaos on that previous slide, just thousands of spreadsheets and thousands of beetles. How do you turn that chaos into something calm, and how could we use the resources at our disposal to help us do the work faster? And so as this slide says, with a program called ArcGIS, which is a geographic information system, kind of like what you can find on Google Earth or other geographic information systems, we can take the hundreds of databases and all the records and turn it into a visual tool that can be manipulated and queried to our liking. And it's kind of like building a virtual Amazon Rainforest video game for scientists. Now,
cool.
And now, the question is, what can we use this digital world to look for? We're not looking for treasure, per se, but it's kind of like treasure to us.
Bug treasure.
It's bug treasure. So now we know that Terry and CJ collected all sorts of insects and measured the trees.
So again, here's our plot.
These
are our two plots.
In Ecuador. So
Ecuador.
Here's Ecuador, and on the eastern coast, or eastern border of Ecuador, you can see where our plots are. I guess next slide. Real quick. And you can see here's this program that we use, we can have an infinite amount of zooming capability. So with the right layers, we can even zoom down to an individual tree if we had the right imagery, satellite imagery, to see that.
So here's our impact plot. This is Onkone Gare, which is the Guarani word for piranha. Mm-hmm. And then this is the control plot to Bettini.
What kind of information, I'd like to ask you guys, do you think we could look for in the data? And so at this point, I guess just feel free to type in whatever type of information you think that we could be looking for. So as we keep zooming in, this is, as you see on the right, is the flat map that we showed earlier of the transect, and on the left is the plot that is being developed in the GIS software.
It's the same transect with the same data points.
Yeah, and you can see that the accordion style that Terry set up, the 10 transects with all the 10 sheets, will be slowly emerging as we zoom in closer and closer.
So this is a close-up of two different transects. Here's one transect, and here's another transect.
And now individual items are beginning to develop here. What do you think that these individual items are going to turn out to be?
Yeah, so what are these green dots here?
What are these green dots? I guess I'll give you a second or two to answer.
Well,
Anybody have an idea?
We're waiting for answers on the green dots. They're starting to come in. We did have a couple of people saying that maybe you could be using this data to look for the distribution of species, the amount of diversity, the niche partitioning. You're finding the answers to the green dots include things like density, bug clusters, species of insects.
I actually see an answer from one of our interns. That's kind of cheating. Yeah, you're right, Mackenzie. These are the trees. So all of these green dots are trees. And those are all great ideas of ways that we could use this database. By the way, all of these green dots are the trees that were georeferenced and measured. The diameter at breast height, or DBH, was measured, and they were identified by one of the experts of tropical botany, Niles Pittman. Nigel Pittman. Sorry, Nigel Pittman.
Duke University.
And so, with all these trees that we have, I think on the next slide, let's see what we got. And here we've overlaid all of our collecting sheets within our transects, and you can see that these white sheets are represented here, just as in the flat map. But as we get further and further, you'll see what the tools of this is. Now here is an image of, if we want to know what the name of all the trees are, or if we want to know what the species name or the genus name or even the height or maybe the diameter at the base height, we can select those attributes out and say label it accordingly. And here you could see that we've just selected the genus names of all the trees in the transect, and you can see that here that brings up that information for us when it's needed. Okay, so now if we have a couple problems that we face besides all these species being new to science, and not only do we not know what to call them at first, but we also don't know their life history or maybe even their habitat preference. And using this tool, we can look for patterns in these data in a simple way while letting the computer do all the work for you. And let's say we want to find where species beetle or morphospecies 21, which is this little
morphospecies 21.
Cute guy right here, where it resides and where it was found. We can build a search to highlight all these sheets that morphospecies 21 was found on. And as you can see in this slide right here, you can see these pink dots represent all the sheets throughout the collecting times where it was found.
So Michael, this is pretty amazing. Earlier Terry talked about if you wanted to go back and find a living version of the beetle named after Arnold Schwarzenegger, you could go back to this map and actually figure out where the likely residence of this beetle might be.
Could go exactly to the same tree.
Yeah.
It's like an address book, a phone book of beetles.
Yeah, exactly.
Pretty amazing.
Definitely. It's using the technology to make it easier. We can also make it draw a 5-meter buffer around these areas, around the sheets, that'll encompass and isolate all the trees.
That's what these pink circles are.
That the green dots residing in the pink circles will be the trees that are the probability of maybe where this beetle came from. And you can see here we've isolated them out and they've turned into red dots. Let's see. And so we've already had this set up for you, but in real time, I mean, it would be almost just as fast, and in a matter of minutes we can start to see patterns of possible trees that the beetle might live on, either feed on, or possibly lay its eggs on. And now, if we click on one of these trees, like CJ just did, you can see that the tree
genus Maltesia.
Maltesia
Malacocalix.
Malacocalix. It's found to be a very common tree within the buffers around where the morphospecies
Here's all the
21
occurrences
resides.
of that genus.
And you can see that we've highlighted them here in this database. And so how do you think that we can test our hypothesis that beetle 21 possibly lives on the tree of the genus Maltesia? I guess you could type in your answers now if you want.
What would you guys do if you wanted to test this hypothesis?
So you're looking at the data, and the data is suggesting that this particular bug might live on a particular species of tree. So how do you
have correlative data.
So how would you test that? That your data modeling is accurate.
And let's assume now you have unlimited funds and unlimited time.
So Chris or someone at Coffee Middle School is saying set up more stations around those types of trees.
Okay, it's an excellent idea.
Lisa says you could look for that beetle in other trees.
Okay, those are all excellent ideas. We could also go back down to the Amazon and find that individual tree, because remember we have all the trees mapped, and we could actually climb up into the trees and try to find direct evidence of that beetle in and around those structures on that tree. So maybe it's pollinating that tree, maybe the female is laying eggs in that tree, maybe the larva lives in that tree. We really know very little about the natural history and the life cycles of these beetles in the Amazon canopy.
But we do know the address.
We do know their address. That's right.
But sometimes, you know, scientists don't have all the funds to go back down to the Amazon and spend another 6 months in the forest. So another project that CJ has been working on is taking the gut contents out of the insects and what they eat and synthesizing the DNA and trying to match it with already known plant DNA. And it said that we would have to spend less time in the Amazon, but we can get a lot more accomplished with the technology and the proper records and databases that was originally collected by CJ and Terry.
So we're testing this out right now. I'm working with some colleagues from our botany department who are trying to they have developed a DNA barcode for plants, which is like a DNA fingerprint, a way to identify plant species from just a fragment of leaf material or fragment of a root. And so we're testing this out on specimens that we collected in Maryland, and we're finding that we can take the gut contents of the beetle and sequence the DNA and match it to their plant database. It's kind of like a fingerprint database. But the problem is for using this in Ecuador right now is that we don't have a database of DNA sequences that map to reliably identified plant names. So it would be like if you were on a crime scene and you collected fingerprints and you matched that set of fingerprints to fingerprints in a database, but the database didn't have any names associated with it. You'd know that the person was at the crime scene, but you wouldn't know what their name was. So you could do something with that data, but not a lot. So what we're going to try to do in the future is develop a database of DNA, a DNA barcode reference library for the plants and the beetles, and see if we can then use DNA to associate beetles with trees. Okay, so just want to, in the last few minutes, I want to throw out some thought exercises for you guys of how you can take what some of the problems that we've been trying to solve in the Amazon and apply them to your backyard. So I want you to think about how many kinds of trees are in your backyard and how many individual trees are in your backyard. Do you have trees in your backyard or the closest park? Okay, remember the kinds of trees is the richness and the number of trees is the abundance. And when you combine those two together, you get the diversity statistic. So diversity is a combination of species richness and individual abundance. And so if you look at all of our Smithsonian forest plots around the world, and this is the link where you can see all of them, I just picked two examples here to compare to. So this is the plot that is near our plots in Yasuní, Ecuador. This is a 50-hectare plot, so a lot bigger space. And you can see how many trees, individual trees, and how many species of trees they collected. And compare that to a plot in Wisconsin. And it's not really fair because the Wisconsin plot is half the size, so if we just cut the Yasuní numbers in half, you can still see that there are 557 species versus 36 species. And so the Amazon rainforest is just so much more, has such a higher species richness of trees and a higher individual abundance of trees per hectare. So another homework exercise for you guys is to go and look in our book chapter, and we have a list of how many tree species are in our Onkone Gare plot and in our Tipitini plot. And remember, these two plots are only 1 hectare, so 1/25 the size.
Thanks everyone for sharing some of the statistics that you already know offhand about your own environment.
Yeah, so we're getting some answers in from people. So this is something that you can do with diversity data. You can calculate what's called beta diversity, or change in diversity across time and space. So this is just showing you the numbers as they get closer to 1 mean that the two plots that you're comparing are identical to one another. And you could do this with any two plots in space. So at the family level, at the coarser taxonomic level, the two plots, the transects within each plot, overlap by between 60 and 70%. As you go down to the species level, that goes down to about 20%. So that means that the transects have about 80% unique tree species in them on average.
This is pretty amazing.
In Ecuador, right.
So in those small, what was the size of
1 hectare.
In 1 hectare, there's only 20% overlap from hectare,
Right.
from sampling to sampling.
Right.
And for some people, maybe we just translate the hectare. That's 2.2 acres.
Right. So what we're trying to figure out is how what are the patterns in the beetles? Do the beetles map to the patterns of the trees? And so the last thing I'm going to talk about is the group of beetles that I've been working on, the tumbling flower beetles in the family Mordellidae. And so this is a sample of just some of the beetles that we got just from one sampling sheet. And you can notice the diversity in size and shape and color of these beetles. We know that they're pollinators. We don't know exactly what they're pollinating, if they're specializing on any one tree or if they will pollinate every tree. This is just a few second clip of why we call these the tumbling flower beetles.
Okay, we'll go ahead and bring this up
Yep.
for everybody.
This is our Mordellidae gymnastics. Not doing anything. Touch them. Do something. This is one of my interns from last summer. This is Donna.
Oh, nice.
You can see that's the tumbling behavior and why they're called the tumbling flower beetles. And so what I'm trying to do is look at these beetles at a species and genus level, again a finer taxonomic resolution, and understand how they're arrayed in space, across space and time, and if this matches the trees, and then if this information is useful for conservation. So here's some of the prettier and noteworthy beetles, tumbling flower beetles of the New World, and I think they kind of look like some of our noteworthy gymnasts from the U.S. I'm a gymnastics fan, so I like to compare the two. Okay, and so I did some statistics on these guys, and so this is the same statistic from the trees. This is just taking the kinds of trees, and you can see that as you look at just the common beetles, so the ones where we found at least 50 individuals, the transects are almost the same as each other. So that means that the beetles that are common not just locally in one sheet, they're common across both transects. But we also have many beetles that are rare, and we're only found 1 or 2 individuals. And so if we look at the other statistic, which takes into account richness and abundance, you'll see a slightly different pattern. The bars go up, but they don't go up as much. So some of these beetles, even though they're found across the plots, are more abundant in one transect and less abundant in another transect. And so this is an ongoing project, and I'm trying to understand why this is and how the pattern of beetles maps to the pattern of trees. And so I'm going to leave you with some questions about why this might matter for conservation. You know, if we were able to preserve every single hectare of rainforest, maybe it wouldn't matter, but we can't preserve every hectare of rainforest. So how do we choose which hectares to preserve and which hectares to use for different purposes? And so what I'm trying to do is come up with a model to compare any 2 patches of rainforest and understand how much overlap there is in biodiversity. How different are they from each other? And then the very last thing that I'm going to talk about is how we go about naming these new species. And so this is the same family of beetles I was talking about, and all the different ways that I'm going about trying to put names on these beetles. I'm trying to work on a world catalog. I'm trying to build new keys so we can identify these. We're going to be making Encyclopedia of Life pages, which our director, Dr. Samper, talked about in the intro welcome message. And then doing some new species descriptions. And so this is a description I've actually been working on with our newest Natural History Museum junior taxonomist, and his name is Jack Mills from Williamsburg, Virginia, and he helped me describe a new species of tumbling flower beetle. And this was the species that was originally collected by an Italian priest, and he sent it to his colleague over in Italy, and they found that there was fungus in the guts. They called it the fungus-eating tumbling flower beetle. And so he predicted, Franciscola, when he described the species, that they would find more individuals if they looked in the canopy. And sure enough, he was right. So this is the known species on the left, and these are 2 of our new species that we're describing. And this is the species that Jack's helping me describe. And so you got a picture of him.
CJ, not only do you have a picture, but Jack is online with us today.
Oh, hi, Jack.
And in fact, he pointed out this was indeed the beetle he helped measure for his science project. There he is.
And so we had a great time. Jack came over to the museum in December, and he spent all day measuring this beetle and the different attributes and structures of this beetle and helping me determine that it is a new species. And we even come up with a name for it. And we haven't published it yet, but we're working on it. And so it'll be hopefully published soon.
Great. Thanks for joining us,
So
Jack.
yeah, I want to thank Jack for all his help, and he did a great job with his science fair project last month. Okay, and so I'll just leave you with some questions to think about. You know, why do you think insect collections are important? We think they're very important. They're the foundation for everything that we do at the museum, having a reference collection to compare things. There are 2 reports that were published recently by the National Science Foundation and the White House Office of Science and Technology Policy, and there's a lot of great information in those 2 reports about why biological collections are important and why we need to fund them and preserve them and make sure that they are fully staffed.
And I'd just like to leave everybody with something very confusing and difficult to think about. Remember that we said that there's 100,000 species or more per hectare and 3.2 times 10 to the 10th individuals per hectare. The Amazon basin has 17 billion hectares, and there's at least 450 different kinds of forest in the Amazon. So, as CJ says, this plot versus that plot, what's the beta diversity? And we have to do it for 17 billion hectares.
Terry, that has to sink in, and I'm not sure it ever can for any of us. We finished yesterday's session describing how we can grasp the vastness of the universe, but somehow I have a feeling grasping the vastness of the number of species just in this one part of our planet, it might be just as difficult for us to understand. Certainly without the help of your guidance, it would be almost impossible.
Well, it's a very interesting thing that you said, because actually what we're dealing with is fractal universes. So we've got the universes, our universe down into our galaxy, down into our solar system. And now what we're getting into is to the smallest fractal universes. And remember, one of CJ's little Mordellidae beetles has a whole bunch of gut parasites. It has little things living on its surface. And so you keep going finer and finer and finer ad infinitum.
Pretty amazing walk through the Amazon and through the universe, as you put it, really, Terry. I mean, it's pretty amazing. I want to thank all three of you for joining us today. I think we have one final photo here to bring up of the team, the Canopy Beetle team.
Yep, this is just part of our team. There have been many other students working on the project through the years.
There are people asking how they can help.
Yeah. Well, actually, that's a great question, and I want to take this time to thank all of our interns. These are all of our volunteer interns who have come and helped us. Amelia and Silvana databased all of the beetles, and the other interns, Maggie and Mackenzie, who have been working with us the past few months, have been imaging all of them. So if you want to come and do a volunteer internship with us, get in touch with us, and just go and look in your backyard and see what kind of diversity is out there. You know, if you can start your own insect collection, then and you can find out how to get those data to your local entomologist or your local museum. The more data we collect, the better answers that we can give decision makers, I think, for conservation problems and conservation questions. So.
Well, thank you all for your great work and for sharing it with us and for concluding our conference on problem solving. You've certainly laid out what the problems are and how we can begin to look at them in some really amazing ways. I do want to invite everyone to stay on with us. I would like to turn the floor over to Stephanie Norby, who's the director of the Smithsonian Center for Education and Museum Studies, and I'd love for you to listen to some closing thoughts and share some of your own closing comments with us. So here is Stephanie Norby.
I hope you enjoyed this introduction to just a few of the problems that experts at the Smithsonian are investigating. If you missed a session, they'll all be archived on this site. And to continue to explore these problems, we hope you visit the exhibit hall. I want to thank Learning Times, who are our valued partners in producing this exhibition, in producing this online conference, and to Stevie Engelke, who is the impresario behind the curtain that pulls it all together. And of course to all of our Smithsonian experts, who not only for their presentations, but for all of the amazing work that they're doing in solving these important problems. And finally, to Microsoft Partners in Learning, who sponsored this conference, and without them it wouldn't have been possible. So now that you're part of the Smithsonian family, we want you to stay in touch. Stay tuned for some upcoming announcements about other ways that you can get involved, and we look forward to continuing these conversations.
Thank you, Stephanie. We encourage you to continue to share your feedback and thoughts. We have putting up a feedback link for an overall conference evaluation. We'd love to hear your thoughts of the overall conference, but we'll email this to you as well in the next day so you can do this at your leisure. One more thank you, if I may, and I'd like to give you a glimpse into some of the virtual illustrations that have been collected. Dan Porter has been busy capturing your contributions as well as the contributions of our Smithsonian experts who are with us. And so do check out some of these fantastic illustrations, including this one, which is from our Part 1 series of sessions on what can clothing communicate. And we'll be posting these illustrations for you with each program description. And if you attended these sessions, I think you will find it a great way to remind yourself of the journey we took together through each of these topics. So I'll flash these across the screen as we wrap up, and remember our time spent together throughout this conference. We'll see you all online soon. Again, thank you, Stephanie, and thanks to the entire team that put the conference together. Take care.